The overall goal of this experiment is to detect enterohemorrhagic E. coli colonization in mice using a non-invasive in vivo imaging system.
This method can help answer key questions in the enterohemorrhagic E. coli pathogenicity field, such as colonization. The main advantage of this technique is that EHEC colonization can be detected in living animals without sacrificing animals.
To begin, streak a negative 80 degrees Celsius stock of E. coli 0157H7 EDL933 bacteria, harboring a luciferase plasmid onto an LB agar plate with 50 micrograms per milliliter of kanamycin. Grow the bacteria for 16 to 18 hours at 37 degrees Celsius. Pick a single colony from the overnight plate and inoculate 3 milliliters of LB medium with 50 micrograms per milliliter of kanamycin. Then, incubate the culture at 37 degrees Celsius and 220 RPM for 16 to 18 hours. The following day, prepare 2 milliliters of a 1 to 100 dilution of the overnight culture and inoculate it into 200 milliliters of LB with kanamycin. Incubate the culture at 37 degrees Celsius and 200 RPM for 2.5 to 3 hours, or until the optical density at 600 nanometers reaches between 0.9 and 1. Next, centrifuge the recultured bacteria at 8,000 times gravity and 4 degrees Celsius for 30 minutes. Discard the supernatant without disturbing the pellet and wash the bacteria with 100 milliliters of